Hey guys, welcome back. We're still in chapter 7, which is dealing with multifactorial traits. And today we're going to be looking at section 3, methods used to investigate multifactorial traits. So we're going to talk about some of the terminology associated with those and then how we can study these traits where environment and genes play a role. So let's get going, shall we? So the first thing we're going to talk about is something called empiric risk. Now, first thing to know that empiric risk is not a calculated value. It is written as a percentage, but it's based more on the observed population statistic. So it's looking at the percent of recurrence, meaning essentially the likelihood of someone having a particular disorder or trait based on different degrees of relatedness. So empiric risk is used to predict recurrence of a multifactorial trait in a family. It increases with the severity of the trait, so the more severe a trait is, the higher the empiric risk is, meaning the more likely someone is to get it. The number of affected relatives, so the more people in the family who are affected, the more likely they are to be affected as well. The sex of the family members, depending on if it's a sex-linked trait, and the increasing relatedness to an individual, to an affected individual. So what we mean by that is, as you see at the bottom, we have this chart here, empiric risk of recurrence for cleft lip. So they're looking at the likelihood of someone having a cleft lip based on cases of it in the family. So if an identical twin has a cleft lip, then the person has a 40% chance of also having one. If a sibling, so non-identical twin, any other kind of sibling, has a cleft lip, then there's only a 4.1% chance of getting it. If their child has it, then the parent has a 3.5% chance. If they are the child, so if their parents have it, sorry about that. If their parents had it, if they are the child of parents who had it, then they have a 3.5% chance of inheriting it, because this is the relationship to the affected person. So if they're aunt and uncle, so if they are the niece or nephew of someone with a cleft lip, they have a 0.8% chance of getting it. If they are a first cousin to someone, then they have a point of someone who has a cleft lip, then they have a 0.3% chance of developing it. And general population risk, meaning that no one in their family history, no known relatives have a cleft lip. There's only a 0.1% chance of them developing it. So you can see how, in this case, someone who is genetically identical to them, that still doesn't mean that they will necessarily both have the cleft lip. It's a very high likelihood. 40% is still much higher than the rest of these numbers. So that indicates that there is some genetic factor involved, but it is not a guarantee. And then as we get farther away, being siblings, child to someone with one, being the niece and nephew of someone with cleft lip, as we get further away in terms of relatedness, then you see the percentage, the likelihood dropping off. And here's another one. This is for schizophrenia. So if there is no family history of schizophrenia, then a person will only have a 1% chance of developing it. If their family member, if they have a sibling who has schizophrenia, then they have a 7 to 10% chance of developing it. A dizygotic twin, meaning a fraternal twin, then the risk increases to a 10 to 18% chance. Monozygotic twin, or identical twin, then it increases to a 40 to 50% chance of the other person developing schizophrenia. If one parent has schizophrenia, then the child has a 7 to 16% chance of developing it. If both parents have schizophrenia, then there is a greater than 50% chance, or greater than or equal to 50% chance of the child also developing it. If one parent and one sibling both have schizophrenia, then there's a 15 to 20% chance of someone getting it. And if a second degree relative has schizophrenia, so no one in the immediate family has it, but maybe a cousin or something like that, then there's only a 2 to 5% chance of someone developing schizophrenia. Again, you can see how the values changed. Now let's move this, shall we? So, heritability. Heritability estimates the proportion of variation in a population for a multifactorial trait that is due to genotype. So heritability is looking at the different expressions of a trait based solely on the genetic aspect. So even though this is a multifactorial trait, and we know that the environmental factors will affect it, heritability tries to figure out just the genetic contribution. So there's something called the coefficient of relatedness, and this will indicate the proportion of genes that relatives share. So if you have a higher coefficient of relatedness, then it means that you share more genes with someone. And that's what the next bullet point says. The coefficient of relatedness increases as more closely related individuals are compared. So if I'm comparing me to my sister or to my brother, we would have a high coefficient of relatedness because we're going to have quite a few genes in common because we have shared parents. If I compare me to a parent, it's going to be a small or a different coefficient. It won't be as, it will actually decrease a little bit from when I compare me to my sibling. And if I compare me to my grandparents, it'll decrease even more because now I'm getting further away in relatedness. So the coefficient of relatedness will decrease as well. Studying comparing multifactorial traits in humans is difficult. A lot of that is because, one, as we mentioned, the environmental component, but also we have to pull the data from many families because we have to look at how it is in not just an immediate family, but also, first cousins, second cousins, third cousins, etc. And like great grandparents and great aunts and uncles, because when we develop things like that empirical risk percentage, we need to know, how many people in the overall family have it and to what degree of relatedness there are. So it does get difficult to collect data from so many different sources. Analysis of multifactorial inheritance has widespread application in agriculture. So what this means is we can kind of look at how some of the traits, if they have maybe a lower heritability, so lower genetic contribution and more environmental contribution, then we can, such as diet, the diet that you take in, then we can change our agricultural needs. So we can grow certain plants that have more of the vitamin minerals needed in our diet to either prevent a disorder or promote a healthy trait. So again, that's looking at how much of the variance is based on the genes versus the environment. I'm moving my camera. Put this back up here. And then we'll come to here. So this is just looking at comparing heritabilities. So if you want to see the entire thing, heritabilities for some traits, for some human traits. So this is a trait. These are the heritabilities. Now, specifically, we're looking at the coefficient of relatedness and all that. So we're comparing the actual proportion with the predicted one. The heritability, if it is a 1, it indicates that under particular conditions, the phenotypic variability is genetically determined. So the closer you are to 1, the more genetically controlled it is. So clubfoot has a heritability of 0.8. So that means that there's a large genetic influence, but a small environmental one. Height, 0.8, again, large genetic influence, but as we already said in the previous presentation, diet and health can affect your height. Blood pressure, 0.6, and now we're kind of seeing it drop a little bit. Body mass index is only 0.5, so that means that there's a larger amount of environmental factors, such as our diet or exercise or lack of exercise that affect that. Verbal aptitude has 0.7, so a little bit higher. It means it does have a strong genetic component, but also definitely some room for environmental influence. Mathematical aptitude is actually only 0.3, so it doesn't have a strong genetic component, but a much stronger environmental component. Spelling aptitude, 0.5. Total fingerprint ridge count is 0.9, so it does have that large genetic factor, but a slight environmental factor, which we mentioned is the heat, mom's body heat during pregnancy. Intelligence varies between 0.5 and 0.8, so there is a good to large genetic component, but also a clear environmental influence as well. Total serum cholesterol, 0.6. All right, let's look at the coefficient of relatedness. So what this is looking at is proportion of genes shared between 2 people related in a certain way. So we have relationships, the degree of relationship, and then the percent shared genes, and in parentheses is the coefficient of relatedness. So sibling to sibling is a primary relationship or a first degree relationship. They have 50% shared genes. Their coefficient relationship is 1/2. Parent to child, again, degree of relationship, 1 degree. 50% shared genes, coefficient of relatedness, 1/2. I have seen that value a little bit different in other charts, but regardless. Uncle or aunt to niece or nephew, that's a second degree relationship or secondary relationship. Percent shared is 25% genes. Coefficient of relatedness would be 1/4. Grandparent to grandchild, second degree relationship, 25% shared or 1/4. First cousin to first cousin, that's a third degree of relationship. And that would be 12.5% or coefficient of relatedness of 1/8. I start talking about some of the studies that we do to look at and compare genetic versus environmental influence. So the first one is an adoption study. So what adoption studies are is you can compare the traits that are seen with an adopted child compared with their adopted parents and their biological parents. So to assess the influence of heredity and the environment, traits of an adopted child can be compared with those of the adopted and biological parents. Meaning, if I'm looking at the child and I notice that they end up having some heart disease and high blood pressure and all that indicators at a young age, I can compare that by looking at both sets of parents. The adopted parents have healthy hearts and they have a good healthy lifestyle, and they have the child go through a good healthy lifestyle. But the biological parents, both of them showed high blood pressure, high cholesterol levels, and early signs of heart disease as well. So the fact that we see it in the biological parents, but not the adopted parents, tells us it's more likely to be a genetic cause. And the reason for that is the adopted child is living in the same environment as the adopted parents. They're eating the same foods, they're exposed to the same conditions. But since we don't see the same trait, that tells us it's a very likely to not be as much environmentally influenced as genetically. Both parents had the same condition, but they live in very different environments. So we tend to consider that a stronger genetic component. However, if we reverse that, if the child has the early signs of heart disease, high cholesterol, high blood pressure, etc., and so do the adopted parents, but not the biological parents, then that means it's probably something more in the lifestyle. It's a stronger environmental influence, because the adopted child and the adopted parents, they don't have any shared genes, but yet we're seeing the same trait in both groups. So that means it's more likely to be caused by something that they're exposed to, whether it's their diet, exercise, lifestyle, health, etc. And that's just kind of what this is demonstrating. Here we have our adopted child. They have shared genes with the biological parents, but not a shared environment. They have a shared environment with the adopted parents, but not shared genes. So by comparing the traits among these three groups, it allows us to kind of narrow down what's the bigger factor, a genetic cause or an environmental cause. Another type is called twin study. So we can also use these to kind of separate genetic and environmental contributions as well. And that's by looking at the frequency of a trait or the likelihood of recurrence in different types of twins, meaning in monozygotic or identical twins versus fraternal twins or dizygotic. So what is the likelihood that both twins will have a certain trait? Do we see that the identical twins, it's much more likely for both members of the identical twins to show the same trait, and less likely for both members of the dizygotic, which would then indicate a stronger genetic component because identical twins have the same genes. But if it's pretty even, it doesn't matter if they're identical twins or fraternal twins, if we see a pretty even frequency of both members of the pair getting it, then it's probably more likely something in the environment. The concordance of a trait is the percentage of pairs in which both members express the trait. So what that means is, concordance is just simply us looking at the likelihood that both twins will have a trait. So if you see a higher concordance in identical twins than you do in fraternal, likely a stronger genetic component. If you see equal concordance in identical and fraternal, then likely an environmental component. There also have been cases of monozygotic, or again, identical twins being separated at birth, and then we can kind of compare traits in their adult selves. They've been raised in completely different environments, and maybe they didn't even know they had a twin, but we can still see certain similarities in tastes and choices and all that, which gives some pretty interesting insights to the idea of nature versus nurture and that whole debate. But that's a whole other issue. So this is just kind of reminding you the difference between identical versus fraternal twins and why they could have different concordance. In identical twins, we have one egg being fertilized by one sperm, and then as it's going through mitosis, splits into two clumps of cells that are genetically identical and develops into two babies who are genetically identical. So they have 100% all the same genes, as opposed to fraternal twins, which are essentially just regular siblings that happen to share the uterus at the same time. You have two separate eggs fertilized by two separate sperm, and they develop into their own unique offspring, their own unique child. So they don't have any more or less shared genes than any other type of sibling, with the exception of identical twins. So since identical twins come from the exact same genetic material, and fraternal twins, there might be different combinations of alleles there. Stronger concordance in genetic or in identical twins means stronger genetic contribution. Then we have something called association studies. And these look at SNPs. Remember those? We've mentioned them like how many times at this point? So single nucleotide polymorphism mapping, so SNP mapping, can be used to identify multiple genes associated with a polygenic trait. So remember, SNPs are these specific loci or locations on the genes that we can kind of track and follow and look at. But what we're saying with these is we're using these to try and figure out which genes can have an effect on one of these multiple gene traits, these polygenic or multifactorial traits. So SNPs are useful in association studies. These are also known as genome-wide association studies, because where SNP patterns of two populations may reveal disease-causing genes, susceptibility genes, or guide therapeutic approaches, meaning we can look at the populations or different populations. We have noticed that certain diseases or disorders might be more prevalent in one population versus another. So what we can do is use these SNP studies, and we can look at certain locations in the genome within these two populations and try to identify, all right, are any of these locations, do we see them more in this population where a certain disorder is a lot more common? If so, then it might mean that those locations are the genes that could affect and lead to this disorder. And once we know that, we can then maybe work on some gene therapy. Once we know what is actually going on with that particular gene, we might be able to come up with some gene therapy treatment options to help, I don't want to say cure, but treat that disorder. This is just a picture showing you a blown up picture of the other one. So patients versus non-patients, we take the DNA samples and then we compare them. So these different little dots each represent different locations in the genome. And you'll notice that the different combination of dots and colors are different here versus here. Some of them are different, not all of them though. We have, so by looking at these and trying to figure out which ones are the same versus which ones are different, it could help us to maybe possibly identify which of these sections are associated with the likelihood of different disorders. All right, so that is section 3 of chapter 7. I hope this was informative. Until next time, you guys, stay awesome, stay curious, and stay safe and healthy. All right, guys, take care.